Hello.
Good morning, good afternoon, I think, for you already.
Y- Yes, this is good afternoon for Indian audience. Um,
Yes,
how are you, Dr. Wamelink? Pleasure
fine.
to host you. Pleasure
Thank
to
you.
host you.
Thank you.
In fact, this topic, food for Mars and the moon, sustainable agri ecosystem, and especially this word moon, holds up a lot of importance suddenly for us because recently, on 23rd of August, we conquered the moon. We landed
Yes.
on the moon successfully. The Mars rover is still hovering somewhere there, and we're so proud of it. So, nothing like it. We're so excited about anything about the moon. So, this topic comes at the right time, an appropriate time. And so excited to learn about everything that we can do on the moon.
hmm Yes, I watched the landing live. So,
Oh, wonderful. I'm sure this is something of such great relevance for you as well. You have been, we call you an agri futurist, uh, who really talks about everything that we can do with agri on Earth and in the space. So we have still good 3 minutes to kickstart.
yes.
We have a lot of people joining. Um,
hmm
Before that, I'm like, if you could, and of course, I'll take you when we officially initiate this process. I'll officially introduce you to the audience, but just wanted to understand what Dr. Wieger Wamelink is all about. Uh, who is he? What does he do? What has been his journey until now? Please inspire us.
Well, originally I'm an ecologist who studied plant breeding as well. So that's a strange combination. And as a child, I wanted to become an astronomer. And in this project, those three things come together because plant breeding, of course, is important, growing crops, but also the ecology is important because uh, I'll explain later, but we need a closed agricultural ecosystem. That's what we want to build on Mars. And of course, my hobby being astronomer is still there. So that all combines. I'm a science fiction fan, so read a lot about it, and look a lot of movies. Of course, The Martian is one of my favorites. Uh, but yeah, I also do a lot of ecological work at my institute. I work with biodiversity, effects of climate change on biodiversity, and also nitrogen deposition, which is a big issue in the Netherlands. And that again is, of course, related to agriculture. So actually, maybe it doesn't sound like it, but it all connects. And this is one of the greatest projects I think we've been working on in the past 10 years.
Wonderful. So what has been your academic journey like? When did it start? Your master's, your PhD, and everything else about after that.
Yeah, well, I did my BSc at Applied University, what we nowadays call an Applied University. I was a botanist, like Mark Watney in the Martian. Then I did work for a little bit already at university. Then I went back to the university to do my master's thesis as plant breeder, and then I started working. And besides my work, I did my PhD in ecology.
Okay.
So that's
And
why also it took 7 years instead of normally four.
everybody is used to that. You ask a lot of PhD students, they will say that.
Yeah.
And all the blame goes to the PhD advisor. I'm sure
hmm
not in your case.
Yes, of course.
That's what all my PhD friends, and there's so many doctors in the audience today, or PhDs, doctors, and professors. So I think we can officially start. We are about time here. Hi everyone, this is Saurabh Gupta. I'm the co-founder and CEO of Edorer. With me is none other than Dr. Wieger Wamelink, whom we call as a future agri-futurist. The topic today for the Edorer's Masterclass is food for Mars and the moon, sustainable agri ecosystem. And we all are very excited right now about moon. So there are a lot of exciting stuff to be heard from Dr. Wieger Wamelink. Before that, I'll give a brief intro about him. Let me quickly share my screen. There's something which my team has prepared. Let me share that. Okay. Yeah. So I'm sure everybody can see my screen. This is everything about the author today, the speaker, rather. Dr. Wieger Wamelink. He's a senior ecologist at Wageningen University and Research, WUR, in the Netherlands. As senior researcher, he works on the effects of nitrogen deposition and climate change on biodiversity, especially on plant species. He's also one of the caretakers of the nature gardens on the Wageningen campus, and as campus ecologist responsible for the management of the green areas of the campus. In 2013, Dr. Wieger started Food for Mars and Moon with as goal to grow crops and on Mars and the Moon, applying the regoliths and ice present at the Moon and the Mars. Now, this is very exciting. We really want to know because a lot of people said that this is just a topic, or is it really possible? We'll explore that in the masterclass. The research includes the effects of heavy metals present in the regoliths of the growth, effect of cosmic radiation on the plant growth, the application of human feces as manure, and the need to bring worms, bacteria, and pollinators to create a sustainable closed agricultural ecosystem. The research recently expanded to grow mealworms on crops grown on Mars, regolith simulant, as an addition to astronauts' meals and to circulate plant waste. So to boldly grow where no plant has grown before. Absolutely. This is such an unexplored territory, and we're so excited to hear more about it. Over to you, Dr. Wamelink. I'm just stopping to share my screen. The floor is all yours. Please see if you can share your screen. Let me allow you quickly. If you have a presentation, please feel free to share. And I'm putting you on the spotlight.
Thank you. Thank you for the nice introduction, and thank you for inviting me. And I'm trying to share the screen.
Let me see. Yeah, good.
Yeah, and I'll put it in presentation mode. So could you confirm that you could see it?
Absolutely.
But it's not at the first slide. Sorry about that.
We can say
so
it.
close your eyes. Close your eyes.
Yes.
I would say, but
We'll wait for the first screen.
now it stops. Yeah, okay, it's there. It has some big components, so that takes a while to move up. Well, for me it's good morning, but for you it's already good afternoon to you all. Thank you for having me over. I'm going to talk about growing crops on Mars or the Moon. My name is Wieger Wamelink. I work at Wageningen University and Research, and I'm in a position that I don't have to that much. So mostly I saw it a couple of days ago. I saw that India has landed on the moon, which is a huge achievement. The fourth country ever doing that. And I think at the moment you're one out of two. That is because both the Russians who failed lately and the Americans who haven't been there for a very long time. So proven only two countries at the moment that they can do it. So a huge achievement. And you landed there. But of course, there is a reason. There is a reason to go to the moon and later on also to Mars. And I expect people to be back in 2 or 3 years on the moon and later also to travel to Mars. And of course, the moon is nearby, relatively. It's only 2 day travel, 3 day maybe, but Mars is much farther away. And when you want to bring people over there and want to live there, you will have to eat. And you can, of course, ship it in, but it is very, very costly and also vulnerable. Huh? And a rocket may explode, and then you end up with no food. And when you're on the moon, you could get back, but when you're on Mars, that's going to be a big problem. And I don't know who has seen the movie The Martian, but that sure shows what will happen if you have nothing to eat and you end up on Mars. You're in big trouble. So we think that when you want to live for longer periods on the moon or Mars or permanently and do research or even start harvesting minerals, for instance, that you should grow your food at your own site. So that is why. But also, it is exciting. And of course, if you join that big journey to Mars, I think it's one of the biggest adventures of the 21st century is to put a human on the Martian soil so that he or she can live there and also uh, live for a longer period. And in our view, that's impossible without um, growing your own crops, not only at the site, but also during the travel to Mars. And then the second question is, of course, is that possible? And that is where we do research. Now, there are two options, basically, when you go to the moon or Mars, how you can grow your crops. And that is aquaponics, so in hydroculture, or use as much as possible that is available on Mars or the moon. And the latter is what we are researching because we want to use not only the ice that is present on the moon, but also on Mars, but also the regolith, so the soil, more or less, that you could find on the moon and on Mars. So we are only looking into that. Hydroponics is, of course, an option, and that is being researched almost, yeah, all agricultural universities all over the world because we apply it on Earth as well. Our food supply here depends partly on it, but growing crops on the regolith, when we started it, we were the first, but nowadays some other universities are working on it and some other groups, but it's relatively new and lots is still unknown. And you will see that in my presentation because we started off very, very simple. Now, going back to this picture of Brian Versteeg, Canadian, who I worked with in the past, you see a Mars base and you also see a dome where you can grow crops. But there is one thing I do not like on this picture, although it looks marvelous, is that you see that it is done in a greenhouse on Mars. And there are two reasons I don't like it. First of all, the effect of radiation. I'll get back to that later. And second is that at the moment, we are not able to build something like that. On Mars and also on the moon, there is no air pressure or almost no air pressure for Mars. And a structure as this would simply explode because we do not have glass or even artificial glass or plastic, something like that, that can withstand the pressure difference inside and outside. So growing crops in space. We are not the first who are looking into that, I already said, but um, it started already with Skylab many years ago, and nowadays you've got the Veggie program where they produce crops in the ISS. You can see it here. It is a relatively small experiment because this is it, and you can also see it works. And one of the major questions back then when they started it was, what does gravity do with plant growth? It's something you can, well, actually not test on Earth. You can do something, but it's very, very complicated. And yeah, the only real good experiment is on the ISS. And we know it grows. So in microgravity, plants will grow, but we don't know how it will be on Mars or the moon, where it's different again. Now, one of the nice things is they can grow crops and they have already eaten it, so it is safe to produce. It's always something that is important, and you have to keep in mind if you grow it in a different place, in a different situation, using different resources than you do on Earth, you always have to test it uh, in many ways. Is it safe to eat? Does it have the same mineral content? Does it have the same amount of carbohydrates and so on? So testing is key. Now, you can see him, the astronaut, smiling with his food. And astronauts, I know also by Dutch astronauts who told me that, André Kuipers, that one of the things they like is when they get resupplied on the ISS, when they get fresh food, because fresh foods is very good for their mood. So it's very important for their well-behaving. And after a couple of days, the fresh food is gone, and then they're back to their normal astronaut food. And yeah, well, it's nutritional. You can live on it, but having fresh food is very, very important, not only uh, in itself for its nutrients, but also for the well-behaving of the astronauts. So that first big experiment that you saw here, actually, most of the crops had to be shipped back to Earth because the people from NASA wanted to do all kinds of analysis on it, as you may understand, content, but also amount, all those things. But the astronauts, who are fully controlled from Earth, what they do, they liked it that much that they eat too much of that first harvest, so not enough went back to Earth. And I talked to the head researcher from NASA, and she said, well, they were annoyed by it because they didn't have enough material back on Earth, but they could understand it. They liked it that much that they simply ate too much. And that already shows how important fresh food is. Eh, when you are in the ISS, when you are on the moon, but also when you are on Mars. Now, we go to Mars, and of course, the first question is, when you go to Mars, is where to live, because circumstances on Mars are very, very different from Earth, but also very on Mars. So there are spots that are more suited and spots that are less suited for humans to live, but also the simulant or the regolith that we want to use, it's different on each side. So what we did is a little investigation based on data that was collected by GPL in the States based on um, satellite images. We collected those and looked which sides are the best to live on Mars, and we did that from a plant perspective. So we know what plants need, and we know what the soil contains, and we put that together and then came up with a map. What are the most habitable places, and especially when you want to use the regolith to grow crops on. So what you see here, for instance, is the amount of cosmic rays that end up on the Martian surface, and that is an issue, not only on Mars, but also on the Moon, since they do not have, like Earth, uh, shielding from an atmosphere. Mars has a little bit of atmosphere, but the Moon does not have it, and also from a magnetic field that blocks most of the cosmic rays coming from the sun and coming from other stars in the cosmos, ending up on Earth. But on Mars and Moon, that's not the case. So the higher the dose, the more dangerous it is. And that's at least dangerous for humans, but as our research showed, and I'll talk about that later, also for plants. So you see here low dosage and high dosage. Low dosage is blue and high dosage is red. So there is a huge difference. But we also looked at soil components. Here is the cation exchange capacity of the soil. So how much nutrients could be in principle available for plant growth in the regolith that is present on Mars. And we looked at macronutrients like nitrate or phosphate, but we also looked at micronutrients like, for instance, metals that we need and the plants need. We looked, of course, also at perchlorate, and this is recently discovered a couple of years ago that Mars, besides heavy metals, also contains perchlorate, ClO4, and that is a bit of a problem because it's really, really toxic, not only for humans but also for plants. So heavy metals for plants is not that a big issue. They can grow up to a certain level. They just take it up and store, but perchlorate is really toxic. There is one exception of the metals, but I'll get back to that later. Now, all those maps, they have uh, a series from low to high, and you can combine those maps, and that is what we did. And then we came up with a map that shows which are the best sites to live and to grow your crops. Of course, not outdoors, indoors, but still we want to use the regolith. And you see, there is a difference. There are sites that are really unsuited. For instance, around the biggest volcano on Mars, Olympus Mons, which has a high amount of radiations, for instance, and it's also very difficult to land because it's very rough terrain. But there are also sites, especially in the northern hemisphere, that are uh, excellent. You can see also here some of the previous landers. You can see that some of the landers actually landed at sites that are really of interest, but also at sites that are less of interest. But that's understandable because up till now, the landers have landed mostly on scientific, interesting places and not yet on places uh, where we should live or where we could live. But you can see that in the northern hemisphere, especially closer to the equator, uh, the situation is better. There's more ice there, more than on the equator, and also the nutrient situation is a little bit better. So that is why we recommend if you would go to Mars and want to stay for a longer period and also grow your crops there, that you go to those sites. Now you see the Hellas Basin, that's gray, and that's for a reason because it's that deep that some of the radar images cannot be produced for the basin. And that's a bit of a shame because a lot of people think at the moment that that could be one of the major parts where you could live. And that especially because it's so deep that the air pressure is a little bit higher and there may be some moist over there. We don't know yet. But yeah, we don't have the information yet to make a full map of it. So that still remains a question. So lots more work has to be done on this topic. So that is where to land on Mars. And for the moon, there are also differences, but the are small, and that's because there is no weathering, for instance, on the moon. Never has been. Well, on Mars there still is with the winds that are there. There's a little bit of air, so there is wind. And of course, also because Mars passed, you have different types of soil there. The past of Mars is a wet one. There were once large oceans and streams. That I think has been proven now more by all the measurements that have been done by the rovers on Mars and also by the satellites. And we still know that there is a lot of ice subsurface on Mars. So that's a major difference between the Moon and Mars. That Now, when we land on Mars, since the circumstances on Mars and also on the Moon are quite harsh, and we cannot just walk outside or grow crops outside, given that, because, well, on Mars, it mostly is around minus 60 degrees, so 60 degrees below zero, and it can go up, well, actually down to minus 100 during the night. So it's too cold for plant growth outdoors. On the Moon, it's even worse. When you are in the dark of the moon, which lasts almost 2 weeks, uh, um, then it can go to minus 200. But when you are in daylight, it can go up easily to above 100, up to 200 degrees above zero. So huge difference, but also impossible for plants to grow. On Mars, there's almost no air. There is a little bit of air pressure. It's comparable to Earth when you are at 30,000 meters above the Earth's surface. So that's 3 times higher than a commercial plane flies. So there is a little bit of air, but it is really, really thin. Plants cannot grow in it, and we cannot breathe in it. So when we go up out on Mars, you still have to suit up. You can never walk outside without protection. Now, on Mars, the gravity is about 1/3 of Earth, and on the Moon, it's 1/6. And actually, we still don't know what will happen. I already explained how it is in the ISS with microgravity, plants will grow, but we don't know what the effect of 1/3 of Earth gravity or 1/6 of Earth gravity will be on the growth of plant species. Um, we speculate about it, and that's also a bit of fun, of course, to do so. And well, on Earth, the maximum growth of trees is limited by gravity. In the States, there are trees that grow up to 100, 120 meters. The sequoias, that are the biggest trees we know on Earth. And that's limited by gravity. So if you would bring them to Mars, they may grow up to over 300 meters, or on the moon, 600 meters. We don't know if they are capable doing that, but we like to speculate that it is possible. And then imagine that you would have a forest on the moon with trees of 600 meters high. But to build it, to grow it, you need to do it indoors. And well, that asks for a huge structure. So if it is feasible, I don't know. And if we're going to test it in the near future, I doubt it, but it is interesting to think about. Now, a third thing on Mars and on the moon, I already talked about it, is the cosmic radiation. That is a real problem. It is a problem for humans. You have to protect yourself, but it is most likely also for plants. I'll get back to that later. But in our view, you will live in a dome, and if you want to call it a kind of hobbit home, but it will be a dome and it will be below ground to protect you from the cosmic radiation. And I asked people who have more experience in that, and they think it needs to be at least 3 meters thick. So you build your dome and then you cover it with the regolith, and it should be at least 3 meters, but I also heard 10 meters. I hope 3 meters will be sufficient because 10 meters is in itself already a problem. But if you are indoors, then of course you are safe. And what is easy is that you live indoors in normal air pressure, Earth-like air pressure and Earth-like atmosphere. And that makes life for me also a lot easier because I want to grow my crops over here, as you can see depicted here. And that's also going to be indoors in normal air pressure. So I don't have to solve those problems. We are indoors, so I'm going to use LEDs, which also is beneficial. I can mimic every type of radiation I want, light quality, I can play with that because I use LEDs. So the only thing I would like to use from Mars and the Moon is the ice that is present and the regolith. Then still, of course, the question is, will the plants grow on the regolith? Now, the regolith is present both on Mars and on the Moon. So that's there. There is ice, especially on Mars. There's quite a lot of ice. You still have to collect it and to dig for it, but it is there. And of course, on the poles, there is a layer of CO2 ice, so cold ice, but also water ice, and that could also be collected. On the Moon, it's more problematic, but the Indian spacecraft did not land for nothing on the site where it did, because we expect that on in the neighborhood there is more ice, but it still has to be proven, and that's something then you can use. So we'll probably will know because the rover I know today it started driving, and hopefully it will find ice very very soon on the moon, and that would be a next step in going to the moon and living there. Now the Martian atmosphere that is there, it's very thin, as I said, but it contains CO2, which is handy for plant growth. You could use it. There is N2, and there's also a little bit of oxygen. So the most essential parts of Earth's atmosphere is also present on Mars. So you could make your atmosphere for your dome from the atmosphere that is present on Mars. But of course, you can also make it in an artificial way. There are several options. So key is that we have to protect ourselves when we live on Mars or on the moon, and that it will most likely be in a dome. The other option is that you will go and live in a cave. That is also possible because then you're also protected. Now, the first problem I mentioned already several times is the cosmic radiation. And we did several experiments to see what the effect of cosmic radiation is on plant growth. Here you see the experimental setup that we had. We did not do that in Wageningen. We did that in cooperation with the University of Delft. They have a small nuclear reactor, and they can produce cobalt 60, which emits uh, gamma radiation in an amount that is comparable what you would experience on Mars and the Moon. It is a relatively low dosage. It's less than what you would get if you take a picture at the dentist, for instance. But still, you have to be careful, and that's what you see here. You see what we call our castle. So it is shielded from all sides to protect us from the radiation. But the dosage is very low, so you can work with it and work in it for a certain amount. You see Nienke here on the left. She did the first experiment, and that's also published. Um, and the experiment looked a little bit like this. And on the left side, you see very, very tiny, so in an early stage, garden cress plants. And on the right side, you see rye. And those species we often use because seeds germinate within 24 hours, which is very handy, and they grow rather fast. And you can harvest also something in a relatively short period of time. We are not working with Arabidopsis, if that is going to be one of your questions. I know this is one of the plants that is often used for experimental research, but you cannot eat it. So I did not see the point in using that. We really immediately started with crops. That's where you want to go to. You want to work with plants that you actually can eat. You see also a bit of Lego here, and that is especially there. That was of the brother of Nienke, and he was very afraid that he never would get it back. The Lego was used to use a piece of paper, to put a piece of paper in it that collects radiation and that we could take data from later on. So it collects the dosage, you can measure it, and later on you can see what the actual dose has been for the plants. But the plants are growing up. So the idea was that we wanted to measure at the tips of the plants because they get the highest dosage. And then, yeah, the papers had to be raised as well, and that's easy to do with the Lego. But the problem was that everything that has been under the radiation circumstances has to be checked afterwards. And when it's become radioactive, it will be destroyed and never can be used again. So the brother of Nienke was very, very afraid that his Lego would be destroyed after the experiment. But it never was a problem, so he got it back. But that is a simple way of doing something innovative in an experiment. Sometimes the solution is very, very simple. And these are the results. And we saw major effects of the radiation. So the even low but constant dosage that you would have on Mars or on the moon. So on the top left, you see that the leaf is looking strange. It's narrowed. That is one of the effects we saw. That is right. On the garden crest, on the right side, top right side, you see a more or less normal plant on the left side, but a very tiny, small plant on the right side. So we got dwarf growth as a result. And we got all kinds of necrosis on the leaves. And that's something, of course, you don't want. The plants still did grow and produce biomass, but you can imagine if it has necrosis or dwarf growth, the biomass production is much lower. And that's something you don't want because, well, you're living in a dome and it has to be built. And you can imagine that's complicated. It's not easy to build a dome on Mars, even if you 3D print it. So space available for plant growth will be limited, and you want to have your harvest at its peak. And not that it is only half of even less because of the radiation. And this gives us already um, enough information to say, no, you cannot grow your crops in, for instance, a greenhouse, even if you could build it because it's not shielded. It gets a too high dosage of radiation, so you have to do something. So you have to go below ground to protect also the plants, not only yourself. Now, for the rest of the presentation, I will talk about our experiments and our main goal that we have, and that is to set up a sustainable agricultural ecosystem for Mars and for the Moon. And the basic system is depicted in this picture. You see the main elements that we are working on. First of all, of course, you see the pea with the pods. Growing crops. That is our major goal. But growing crops in itself is not enough. You've already seen you need to build something to grow it in, but that is not what we're doing at the moment. We are not looking at that, how to do that. That's for the technicians. But we are looking at that agricultural ecosystem that has to be circular and sustainable and not linear, like, for instance, the agriculture is still at the moment here in the Netherlands, causing problems. And on Mars, there is nothing. So everything is valuable. Also, for instance, the feces of the people that eat my crops. That has to be recycled and has to be put back into the soil as manure. Now, I know there are still many countries in the world that do that, and they are right in doing that because it contains lots of valuable nutrients for the plants. We in the Netherlands and in Europe more or less forgot about that, and there is a reason for that because you have to be careful because there are sometimes harmful bacteria in your feces. For instance, E. coli, you can get really sick. But we are wasting at the moment our feces, and that is causing problems. It costs a lot of money. We flush it away in our toilets. It costs a lot of money to purify the water again, but also we waste it. We waste phosphate, waste nitrate, and that's causing problems in the end in nature. And especially phosphate is not unlimited available on Earth. So uh, I think it would be better to go back to what we used to do here in Europe and to apply our own feces as manure again. On Mars and on the Moon, it is impossible to grow your crops if you don't do that. But there is more, of course. If you have a pea, you don't only eat, of course, the seeds. The rest is waste. And that waste all has to be returned to the soil. Otherwise, you also deplete your nutrients in the soil. And for that, we need bacteria. You can see them here. also depicted, but you also need worms. And I'll get back to that in extent later on. But for us, they are key. And you also need bacteria maybe to get the soil more fertile. We can get rhizobium bacteria over there. They live in symbiosis with the roots of the plants, especially the legumes, so they can take N2 out of the air. Remember, we are going to make artificial air on Mars and also on the moon, which is comparable to Earth. So there is a lot of N2 in it, and they can turn it into nitrate. And then also, I think key, and we have not done any research on that yet, so there's still a lot of things that have to be done. Um, that is fungi, because fungi can acquire nutrients much better and much easier than plants can, and they can share it with the plants. So again, here symbiosis with the plants is important. And also that is something we forgot about on Earth, at least here in the Netherlands. If you go to a field here with crops and you look at the soil, you will see that the soil is not really healthy anymore. So I think we have overdone it here in the Netherlands. We produce a lot of crops, but the health of the soil has gone down tremendously. And if you look at it, you will find hardly any fungi anymore. So they cannot assist in growing the crops. And also the earthworms have almost gone. So that reduces the possibilities of the plants to grow. The only thing you can do nowadays is put a lot of manure on it. And that looks sensible, but it is not. We do not have a fertile soil anymore, and it is starting to give problems. And not only on the arable fields itself, but also outside because of pollution. You need a lot of chemicals to keep the crops healthy, but those chemicals also flush out of the arable fields and comes in streams, which causes problems for the fish, for instance, that are living in there. So this is not only for Mars and the Moon, where it is necessary because there's nothing there, nothing growing, there are no bacteria, no fungi. Uh, so you have to bring everything there and you have to set up a sustainable agricultural ecosystem. But I think here on Earth also we have to go back to that system. And especially here in the Netherlands, we forgot about that. So I will go into more detail in some of the research we've done to make that sustainable agricultural ecosystem a reality for Mars and for the Moon. Now, first about the regoliths. We know a lot about the regoliths on the Moon because we brought it back to Earth, but unfortunately, that's not available for our research. I hope more will be brought back soon so that we can do research on the actual regolith. From Mars, nothing has been brought back yet, so there's nothing here on Earth, and we are already stuck there with a problem, of course. But there is a solution, because already in the 90s, NASA developed artificial regolith, both Martian regolith and moon regolith, and it is sought and found here on Earth. They looked where on Earth can you find soils that are really, really comparable to what you would find on Mars and on the Moon. And we already have now several generations of it. Here you see an example of the regolith we started with that's coming from an Hawaiian volcano. And you can imagine that soil that you find around volcanoes is similar to what you would find on Mars because Mars also has a lot of volcanoes. So that makes sense. And the first generation of artificial regolith came from a volcano. Of course, it was purified, all organic matter was removed. And well, actually, you can buy it via the internet. So if you want to do your own experiment, that's possible. For instance, at the Martian Garden, look it up. You can actually buy artificial regoliths and do your own experiments, like we did. So we have that for Mars, but We also have that for the moon. Now, if you look at the composition, at the mineral composition of what we used in our first experiment, that's the GAC-1A, NASA used it to test rovers and to test suits, to test the equipment in that they sent to Mars, but they also did that for the moon. You see a major part of it is silicium oxide, and that's on Mars, but it's also on Earth. So nothing strange. There's a lot of aluminium oxide in it, but of more importance for us is that there is iron oxide, quite a lot on Mars. That's one of the reasons that Mars is a bit reddish. It has a lot of iron 3, which is basically rust, in its regolith. For the moon, by the way, it's iron 2. That's why it's gray. There is manganese, magnesium oxide, but also needed for plant growth. Also calcium oxide, even potassium and phosphate, P2O5. So in principle, the minerals are there. But this is not what I am looking at, because I am interested in what is available for plants, what is going into plant solution, because plants cannot live uh, of uh, phosphate oxide in this form, for instance, if it is in a mineral form. It cannot acquire it and it cannot use it to grow on. Well, I asked NASA when they delivered me the first batch of regolith, what would happen if you add water to the regolith, and they could not tell me. So it was totally unknown what was the content when you go to the soil solution if you add water to the regolith. So we analyzed that, and here are some of the results. We did that for our first experiment. So you also see here results for an earth soil, and that earth soil, keep in mind, is a soil that is coming from 10 meters deep from the River Rhine. We used that for our first experiment because it's very, very nutrient poor and comparable to the analysis that we did on the Moon and Mars soil regolith. And you can see that here because it's more or less comparable. The phosphate content, the P total content on the Moon and on the Mars regolith is higher, but the question is still, is that available? If you look at it further down the table below, you see phosphate, for instance, and it's almost absent in all three soils. So that is already problematic because the plants need it to grow on. There is a little bit of nitrate, there is a little bit of ammonium, but it's really, really low amounts. Then, if you go a bit higher again to aluminium, it is present in the moon regolith simulant. And I said before that plants can deal with metals, even if they are in a larger amount available in the soil. But aluminium is different. Aluminium is also really toxic for plants, and you can see that it is just at the detection level, but it is there. It is present in the moon regolith, so that's complicating things for plant growth. Another complication of especially the moon regolith is that it is really, really sharp. If you look at if you look at it under a microscope, you will see that it has really sharp points. And if you recall, if you ever have seen it, uh, small movies of the first people walking on the moon, you can see that they're always dirty. And that is because of the moon regolith, and that is because the moon regolith is so sharp that it sticks to everything, also to the suits. That's why they were always so dirty, also to the rovers. And that may cause also problems for my plants because it's sharp and it may puncture the roots. So if you look at this and you're going to use this to grow crops on, you can imagine that the expectations were low because this is really difficult soils to grow your crops on. So our expectations for that first experiment that we did were low. So this is the first experimental design we had. We had Mars and Moon regolith simulants. They were delivered by NASA. By the way, delivered, you have to buy them, and it's not cheap because it's $2,500, $3,000 per 100 kilograms. So it's not cheap. We used a normal greenhouse here in Wageningen. We had normal earth air pressure. Here, remember, we are thinking of growing our crops in a dome, also on Mars and on the Moon with normal air pressure. We added no extra nutrients because we wanted to know what is really possible on those regoliths if you add nothing. Is plant growth possible? We even went that far that we did not use normal tap water because normal tap water contains uh, nutrients for plants. So we used demineralized water to make sure that the growth that we saw was only because of the regoliths that we used and not because of the water with the nutrients in it. Of course, we worked with normal Earth gravity. We had 14 different plant species, which were, of course, crops. We had tomato, rye, a garden cress, and what was the fourth? Radish. We had legumes, and there's that special reason for it. We already saw that there is not much nitrogen, nitrate in the soil, and legumes can, of course, live with the rhizobium bacteria in symbiosis, and rhizobium bacteria can capture N2 and then turn it into nitrates, and in that way manure the soil and also help the plants, of course, to grow. And we had wild plants from the Netherlands, and there was a special reason for it because these are harsh soils. There are not much nutrients in it, and wild plants are often much better suited to grow under harsh circumstances. For instance, we had a species that grows here in bare sand in the dune area in the Netherlands. There are almost no nutrients there. The water holding capacity of that soil is very low. So that was a reason to also select a series of wild plants just to see if they could cope with the difficult circumstances. So that leaves us with three soil types, the Earth control, the Mars soil simulant, the Moon soil simulant, 14 different plant species, and we had 20 replicas. That gives in total 840 pots. Why 20 replicas? Because that is, of course, a huge amount. Normally, you don't do that. But our expectations were very low. And we had 5 seeds per pot. So we had 100 seeds in total per species. And there was that reason I hoped that at least some of the seeds would germinate so that we would see something and that we had something to measure. Uh, Normally, I never would go to 20 replicas, but in this case, we did. And of course, it was totally randomized in the greenhouse. We had 20 different blocks in the greenhouse. You can see here the different soils. They were per block randomized to give a good result of the experiment and to fulfill all statistical requirements. This is a bit how it looked like in the greenhouse. This is part of the experiment. You can see many pots. You can see even the replicas between the wooden sticks. That is one of the 20 blocks you can see. And if you look carefully, you can already see that there is something green. Plants were growing. And plants were growing in huge amounts. And that's something we never anticipated because what we wanted to was follow each seed. So 4,200 seeds, see if they germinate, see if they form roots, see if they form stems and leaves. And we thought that would be germinated. So it was a huge amount of work. This is, for instance, rye. On Mars soil simulant after 3 days, and you can already see that there are roots, and you can even see the stem already emerging. Uh, They germinated within 24 hours, so that was a big success. You can also see that the roots after 3 days are still above the surface. And that is one of the problems with moon, but they will germinate and you will see on, they will grow on, but it is difficult for the roots to penetrate the soil because the density of the soil is quite high. And that was one of the problems in our first experiment. So there were roots, there were plants, but it was difficult for them to penetrate the soil. And after a couple of weeks, the garden cress started to flower. And that was way beyond what we expected in that first experiment, but it did. But if it starts to flower, of course, you want to have seeds. And then you have your complete plant cycle. But garden cress is a cross pollinator. So it needs to be pollinated by, for instance, bees or flies or bumblebees. And of course, we were in a very fancy greenhouse in Wageningen, and it was totally insect proof. So there were no insects in the greenhouse. And that already also learned us, if you want, in that first experiment, if you go to Mars, it is not enough to just grow your plants. You need to bring more. We already came up with the idea that we needed rhizobium bacteria, but you also need pollinators. Now, in our experiment, we solved that by doing it by hand. So we took a paintbrush and then pollinated our plants with the hand, by hand, with the paintbrush, and that worked. But of course, it's very laborious, and that's something you do not want to do on Mars, where you have enough work as it is. So there has to be a solution for that, and our idea is to bring bumblebees to Mars. That is probably the easiest way. The bumblebees can be in hibernation during the trip. You only bring the queens, and when you need them, you wake them up, and then they can start doing their job, pollinating the plants. And we know from research here on Earth that bumblebees can thrive in greenhouses. And they can really live there. That is no problem for them because you can also think, well, why not bring bees? But of course, you have to tend the hive. You can train the people, but you also have to tend the hive during flight. And if you are half a year or longer on the way to Mars, that's going to be a problem. Uh, Bees are even in winter in hibernation always active. They are still active. So that's much more complicated. And what complicates things even more is that um, bees, beehives will not survive in a greenhouse. They will do their job, but in the end, they will not reproduce. So you will lose your hives where bumblebees can live. An alternative is also to bring flies, but they need a special diet of meat. And that is, of course, also a problem because we are not planning on bringing animals. Think of chickens or cows or pigs to Mars or to the moon. Impossible, of course, in a spacecraft with no gravity, but also it's very inefficient to bring animals to Mars because, uh, well, a cow, its conversion factor is only 10%, so you need to put in 10 times as much food in the cow to even be able to use it. Or a pig, they're very, very inefficient. So that is not what we are planning. We are planning that people will be vegetarian on Mars, and that is because it's much more efficient. We need to bring pollinators. But in the end, we were successful, and we already had in that first experiment, we had for rye and for garden cress, we had a harvest. But that was on the Earth control and on the Mars Sol simulant. On the Moon Sol simulant, results were totally different because most of the plants germinated on the moon Sol simulant, but died very quickly afterwards. And that is depicted here also. You see here one of the results. You see on top, you see the total biomass that was harvested. For the three soils, we have in green the earth soil, in yellow the moon Sol simulant, and in red the Mars soil simulant. You see total biomass on top, and then above ground biomass, below ground biomass, and the ratio between above and below ground biomass. And you see it for all 14 different plant species, and on the right side you see the crops. In the middle you see the nitrogen fixes, on the left side you see the wild plants. And what sticks out immediately, and keep in mind that the y-axis is logarithmic, so the differences are bigger than they look like here in the picture, is that Mars and Earth soil give comparable harvest, but that in many cases the Mars soil harvest, the biomass production was even higher than on our very nutrient poor Earth soil. Now the lines between the three soils per species depict whether the differences are significant or not. So if there are no lines, then the differences are not significant. But if there are lines within the species, then the differences are significant. And you can see that there are a lot of differences that are significant, but mostly between the moon soil and the other two soils, but also still between the Martian soil and the Earth control soil. Martian soil giving a higher biomass production. And that was really, really surprising to us. But this first experiment lasted only 50 days, so not too long. And the plants, well, they did grow and we had a harvest, but for instance, the tomato plants we had never gave any tomatoes. It didn't even flower, and the plants did not grow higher than, well, let's say 5 centimeters. So they did grow, but not very high. And that's, of course, understandable with their lack of nutrients. So, first result, beyond everything we expected, but still not enough for people to eat from and to feed the astronauts on Mars. So a lot of things had to be improved. And two major problems, well, actually three, of course, lack of nutrients, but also the regoliths are very, very hydrophobic. So the water holding capacity is very low. And that was really problematic. We had to water the pots twice a day, and even that was not enough. And you can imagine if there is no water, then there is no growth. So that was really problematic. And we think that the aluminium in the moon regolith caused a lot of problem, but also the sharpness of the particles. We saw cork forming on the roots in the moon soil simulant, and that indicates that actually the roots were leaking, and the reaction by puncturing by the plant roots, and the plant reacts to that by forming cork tissue on the roots, had to protect itself. But the problem is that if the plant does that, it cannot take up nutrients in that part of the root. And that also gives then, again, a lower growth in the end, of course. So it worked, but there were some issues. So this is an overview of the second experiment we did. And the only thing we changed comparing to the first experiment is that we added organic matter to the simulant. And here a second difference is that the earth control, and you see it in the middle, in the front row, you see the earth control. On left and right, you have trays with moon soil simulant, and the reddish one, of course, again, are the Mars soil simulants. And you can see here 10 different plant species, only crops sown neatly in rows. The only difference was, that's of really importance, is that we added organic matter to the regoliths. And we would have used the harvest from the previous year, but we used that for all kinds of analysis, so that was not there. So in this case, we used rye, but the idea is that on Mars, you will reuse, circulate all the organic matter that you did not eat and use it for the next generation. And you can already see here that the plants are starting to grow. And sometime later. Here's an overview of the Mars regolith tray that we have, and you can see here, if you recognize it, I don't know, we have leek, but we also have quinoa, we have rye again, we have pea, we have radish, we have beets, we have tomato, we have garden cress, we have spinach. Uh, so all kinds of different plant species, but only crops. And they started to grow, and adding just organic matter helped a lot. Water giving was a lot easier. And we know from here on Earth that the water holding capacity goes up tremendously if you add organic matter to your soil. Something we have forgotten in the Netherlands as well, because the organic matter content in the Netherlands in agricultural soils is very low at the moment, and that is causing problem given climate change and the droughts we've had the last couple of years. And farmers already experienced that they cannot have the harvest they used to. So bringing back organic matter in the soils here on Earth in the Netherlands is a key issue, but also on Mars. It helps a lot. It helped that much that after a while we were able to harvest our first crops. Here you see the radish on Moon Regolith Simulant, but we were also able to harvest tomatoes, garden cress. The quinoa started to flower. Unfortunately, it did not give seeds, but we know now why. We took a variety that is sterile, so the seeds they never could have formed uh, a harvest, but we didn't know that back then. But we could harvest it, and just by changing one simple thing, adding organic matter, uh, made a huge difference. And you can see that here also. This is the average dry waste of the total biomass above ground produced per tray. We try to separate it, and there are also biomasses per plant species. Remember, we had 10. But it was extremely difficult because it became a kind of jungle in the tray. Everything grew through each other, and that we think was also part of the success because the plants actually helped each other. They benefited from each other's presence. And that's why we got a very high yield, not only on the earth control, which was of course expected, which was now uh, organic soil, but the Marsal regolith yielded the same amount of above ground biomass. There was a little bit of difference, but it's not significant. And that is due to one of the three trays that was lagging behind. The other two gave exactly the same amount of harvest as on the earth control. And actually, it has in the experiments afterwards, it was difficult to achieve that. And we think that is because we had all those crops growing through each other and helping each other. So commensalism, as it is called. What we also see is that we had harvest of biomass also on the moon regolith, but it was lower, and it was significantly lower. And we still think, though it improved soil conditions to add organic matter, we still think it is lagging behind because of the harsh circumstances of the regolith. We also had seeds. We harvested seeds enough to do a germination experiment with it of garden cress, radish, and rye. And here you see the results, and you see that for cress, it did not matter if the crops grew on Earth, Mars, or Moon soil simulant. The germination was the same. The same applies for the rye, but for the radish, it was less. It was significantly lower, especially again on the Moon simulant. And you may think, well, 50%. Who cares? 50% is also a lot. But remember, it's difficult to do anything on Mars, on the Moon, harsh circumstances. We only have limited resources, so we want to have it as high as possible. And 50% compared to let's say 90%, it is a difference and it is a loss. And that's something we want to prevent because we want to have it as efficient as possible, especially because space is limited. Well, and then it was time to taste our first crops. And I was reluctant to do so because we knew there were heavy metals in the soil. And in the old days, we would ask some students then to taste them and see what happens. But I'm told that's not allowed anymore. So we had to come up with something else. So we invited Rachel over. She does all kinds of stunts on TV for young people. And we thought, well, then she can also have the first radish that we harvested from Mars and to see how it tastes, but also to see if it affected her. So here she is tasting. It was all recorded on film and broadcasted on the Dutch national TV. And what is the case we now know nowadays is that we grow our crops a little bit slower than a normal commercial greenhouse does. So they are more tasty. There are more secondary plant components in the crops we harvest, so they're more tastier. And radish is, of course, known for its spicy taste. So, and it's growing in the regolith itself. So you have to clean it very carefully because otherwise you could eat the regolith itself. And, well, that's not very healthy. So we cleaned it thoroughly, and the regolith was in there. And I told her, whatever you do, don't drink from that water beforehand. But it was that spicy that she thought, oh dear, I have to do something. So she actually drank from the water. So the thing I told her not to do, she did. But she survived. She's still living and she's still making TV program and still doing stunts. So it is safe to eat the crops. Of course, that's not the way we do it. This is the way we do it. We did a lot of analysis on it. We looked at the heavy metal content in all the crops, and this is one example for tomato. On the left side, you see for some of the metals the content. In the middle, you see it for the moon regolith, and on the right side, you see it for Mars. And then you can see, yes, there is some in it. There is some iron in it, not unexpected, and it's a little bit higher on Mars, giving the high content of iron in the Martian regolith we use. There is some cadmium, there is some chrome, there is some lead, but it is all way below the maximum amount. So it was quite safe to eat. And then we invited people over to Wageningen because to have the first Martian meal based on the 10 crops we harvested, because our research is mostly funded by crowdfunding, and we asked our beneficiaries over to Wageningen to have the actual first Martian meal, and it tasted great. And that is again one of uh, the goals, to have not only a healthy meal but also a tasty, great meal for the astronauts because that influences their well-being. Now, how did we continue from that first successful experiments? We have been doing experiments with the earthworms, and you see an example here from a pilot experiment. And you can see the worm is in it. And what the worm does is it takes the organic matter, even if it is on the soil surface, it takes it into the soil, eats it, mixes it with the soil itself, and then their feces again, mixed with already the soil, is manure for the plants. So it can transform organic matter, dead plant parts that are not directly available again for the plants. It is not manure for the plants, the dead plant parts. It has to be processed, and the worms can do it. What you also can see is that they dig burrows, and that is also important. I earlier said that the water holding capacity of the soils is not great. Even if you add organic matter to it, it still stands on top for a little while before it goes into the soil. And what you see here is the burrows that has been dug by the earthworm, and that helps with water giving because it is now much easier for water to penetrate the soil. And it's also important for the roots, not only for the water, but also for oxygen. Plant roots needs oxygen, and it's much easier also for air to go into the soil so that the plants can take up the oxygen in the roots. So we think that earthworms are key, and they are that important that we did an extensive study on it, and that is what you see here. What you see here is two soil types. We only did this experiment with Mars soil simulant and Earth control, again, organic matter. And we added organic matter to the Martian pots from a previous experiment. And the content was 5 volume percent. So not too high. We would like to go to 10% or even more, but we didn't have that much harvest yet from the previous experiments. And we also added manure. That was the first time that we did it. We added pig slurry because human feces is more complicated. And pig slurry, pigs are very similar to humans. They eat almost the same fodder, and their feces is also comparable to that of human feces, especially when you look at the nutrient content. So we used pig slurry as a proxy for human feces in that experiment. And we added the earthworms, two species, a Caligonella species and a Dendrobena species, to the soil to do the digestion of organic matter and to manure the soil. We had 4 replicas per treatment, and we put it in a water bath. You can also see it in the picture. And that was especially for the worms, because the plants, they are in the greenhouse, and their optimum temperature is, let's say, 20, 25 degrees. But the worms, they like it colder. They like it maximum to 10 degrees. Then they work optimal. So that's why we cooled it. Um, we had it available, and these are uh, worms that you would find in the Netherlands, and they work under relatively cool circumstances. But it was really complicated to maintain this experiment. You can already see that the pots become greenish, that are algae, and it became more or less a big mess, especially in the water bath. So we don't do that anymore, but this was our first experiment. We also learned from it. We now only use worms that can live in more tropical circumstances so that we can have a normal greenhouse without a water bath and that they can live in higher temperatures and do their job. The experiment lasted for 75 days, days, one crop cycle, and we did it with rucola as crop species. And these are the results. And you see again the biomass weight, the above ground biomass weight, dry weight, and you see here the different treatments. And I'll explain a little bit. The first letter you see below every treatment is whether or not there is a worm present. So there is a W if there was a worm, and there is an N if there was no worm. Then the second letter you see is whether or not there was manure, pig slurry present. So an M for manure or no manure is an N. And the third letter is for the soil type. So it's either earth potting soil or Mars regolith simulant. And what sticks out immediately is, of course, the two manured pots. They have a much higher harvest. And that's not surprising because that is what you expect. We know that from Earth, if you manure it, you get a higher harvest. But a bit disappointing was that adding the earthworms did not result in a higher harvest, even though the experiment lasted 75 days. So the worms, we checked afterwards, they were still alive and they even had offspring, because if you add 4 worms to a pot and afterwards you find 6, amongst which 2 little ones, then you know they had offspring. So they are in itself behaving in a way that you would like them to do. And they did dig burrows, we saw them, but it did not influence the biomass production. And that was a bit of a shame because we hoped it would. And we think that this is because it takes a much longer period to really have an effect. And I saw that now. I have an experiment going for 5 years now. It's in one of my offices. And we see now, after 5 years, we see the first soil forming in the regolith. So it takes much longer. And we think that is the reason why we did not see any effect here. But the good news is that the worms can survive and do their job in such a soil. And I have a little movie made from worm activity in the soil. It is a time lapse, and I will start it. And what you will see is on the left side, uh, Mars regolith with garden cress seeds. If you look carefully, you can see the seeds already with no earthworm, so no activity. And on the right side, you already see one of them. You see an earthworm, and you see the earthworm activity. And yeah, there is. What you see now is the earthworms on the right side. You see the earthworms working. You also see that it is wet, and you see sometimes the effect of that on the glass, but you also see the garden cress growing. You see their roots penetrating the soil, and they also form side roots where they take up with the nutrients, but you also see the worm activity. They dig the burrows, helping the plants to grow, but also uh, digesting the organic matter that is present here as well. But if you look carefully, I do not know if you saw it, but the worms also go to the surface to look if there is something to eat. And if they don't find any organic matter, then they also eat the plants. So you have to make sure that you have enough organic matter for them to eat from. So that is important. But this also was published, but you can see that the worms actually do the job, and we can have a look at them even if they are working below ground. Now, another thing we did is look at the nitrate availability in the soil. It is low in the regolith. Here, again, an example on Mars regolith simulant. And we did, well, Stephen did an experiment with bacteria, with rhizobium bacteria. The plants were inoculated, and we looked if the bacteria could grow in the soil and if they really had an effect on the growth. Now, the important thing was, of course, to see if it's working. And here you see the harvested roots. And you see here, if you look carefully, and maybe you can see it when I point it out with my mouse, you can see here the little knobs. And in this, there are the rhizobium bacteria. So they are able to form a symbiosis with the roots of the peas, and they are live. We cut them and we looked at them, and we could see that they were living, so that they actually are feeding in this case, peas with nitrate, so that the peas can grow better and faster. And by this, we can manure the soil with nitrate. Of course, we can also use free-living bacteria for it, and in the next couple of years, we're going to look into that as well. So if, for instance, cyanobacteria will live in these soils and also can take N2 out of the air and turn it into nitrate, and in such a way manure the soil at least with nitrate. So that was also a successful experiment, but we still have to continue that. I had it also with my green beans, which I'll show later on with another experiment. But the control of that experiment, I saw afterwards, also had little knobs with a different type of rhizobium bacteria in it. So they also could do that on the Martian regolith simulant. So in principle, it works. Now, coming back to that successful second experiment where we saw that growing crops through each other worked very well. Afterwards, we don't only did experiments with one crop, monocultures. What we also do here on Earth on our arable land. There's normally only growing one crop. But I kept in mind that we had a high harvest when we grew crops together. And Rebecca did conduct a huge experiment and is going to be published soon because it's accepted by PLOS ONE with three different plant species. We had tomato, we had pea, and we had carrot. And we grew them together in one pot to see if they could profit from each other. So if there was commensalism. And she also inoculated the pea again with rhizobium bacteria, so to make it optimal. And there was also again some 5% of organic matter in the soil. So more or less optimal circumstances. And we did that on earth potting soil. We did that on nutrient poor soil from earth. Uh, and we did it on Martian regolith. And you see here a picture of the results only of the three crops together. And what first took out was that there was a huge difference in uh, crop growth and biomass production. So the experiment did not go as well as we had with some other experiments. But what also came out was that the commensalism, it worked, especially for the pea and for the tomatoes. They grow together. They had a higher harvest compared to tomatoes and peas that were grown in monoculture. So instead of one tomato plant, one pea plant, one carrot plant, we had three tomato plants in one crop pot, or one pea plant, three pea plants in one pot. So there was a higher harvest, and that was for all three soils. But what was also interesting is that for the carrot, they did not like it to be in a pot with tomato and also with pea. And there is one obvious reason for it, because the pea and the tomato did grow in length tremendously, and they were, yeah, uh, more or less shielding the carrot plant so that it did receive less light. So if you want to do this, yes, it works, and you could do this on Mars because circumstances are different, and we think lots of will be robotized. So you have a robot also to harvest your crops, and that makes it a lot easier to do intercropping. So it works, but be careful which plant species you put together, because the carrot really did not like it, and it was overshaded by the other two plant species. But the other two plant species yielded a higher harvest. So this is something most certainly we're going to pursue further, also on the Mars Sol simulant. And then I'm coming almost to the end of my presentation, and that is that recently we started experiments also with human feces, and we started with urine. I go back to that one first. We started with urine. Um, we did an extensive experiment, and we did not use actual human urine, but we used struvite that you can take from urine, and this, in this case, it was taken from human urine. What you do is add magnesium to the urine, and you can add bacteria as well. And then struvite, which is a crystal, is formed spontaneously. You can take that out of the urine and use that as fertilizer. So you have a mineral that you can use and also easily store if you want to, and use it as fertilizer. That is what we use. And what is also handy is that struvite as a crystal is almost pure. So you can have people that, for instance, use medicine. That medicine can end up, or the remnants of the medicine, in the urine. And you don't want to have that in your food system. So uh, even if the astronauts would have to use medicine when they are on Mars, which could happen, we still can use the urine because we can take out the nutrients as struvite. And struvite is magnesium ammonium phosphate, so it contains two of the most important macronutrients that plants needs, but also one of the other important components, magnesium. And you see here two pictures. On the left side, you see a picture in front, a plant that did not get struvite and also not additional nutrients, only a little bit of organic matter. In this case, a green bean, and you can see, well, the leaves are not looking very healthy. They are yellow. They formed one or two beans, and that is it. So it is possible to have a harvest, but it is really, really small harvest, and it's not enough to feed astronauts with it. On the other hand, on the right side, you see the addition of struvite. You see large, very healthy plants with many beans, even on the moon regolith simulant. And actually, this was the first time ever that on the moon regolith simulant we had a harvest that was comparable to Mars and Earth control. Again, Earth potting soil, so the richest soil you could find. And we think that is because of the pH. The regolith, the pH of the regolith is very, very high, above 10. And that is, of course, also a problem for plant growth, especially for phosphate, because phosphate at a pH of 10 is also almost not available for plants. But by adding struvite, which contains phosphate, I think we solved that problem. And then plants really started to grow. So we got a huge harvest. And that was really the first time. And here you can see that using human urine is very, very possible, and that you can get a really high harvest. Of course, you still have to be careful. You have to test it if it is safe, if it is bacterial free and bacterial safe. But that all looked well. So that is one, again, one other option to manure the soil and to use, of course, to reuse the human urine for manure. And that does not have to start on Mars, but if you are half a year underway, my advice would be to store your feces so when you are on Mars, you can directly use it as manure. So you have already a good start with your first plants when you are on Mars. So don't throw it away, don't see it as waste, but see it as something valuable that you can use to actually start your first experiments or crop growth. Now, if you use urine, the next step, of course, is also to use human feces. Uh, and we did already some preliminary experiments with this. Again, we had some famous person over in the Netherlands and Belgium to donate the first manure. That was then sterilized by fermentation to make sure that there are no harmful bacteria in it anymore. And that was applied to the regolith again. And here you see the first example of Tom Waas, that was the person, his poo that was added to the regolith, and we grew garden cress on it, and you see it is growing. Now, to end my presentation uh, is with something I at the start said we are not going to do. We are not going to work with animals, except for, of course, the pollination and the worms. We need them, but not as fodder. Well, then a student came to me and said, why are you not working with insects as food for people, especially mealworms? And you see an example here. This is the lesser mealworm. Uh, they are very, very efficient in turning what they eat into mealworm. So the conversion factor is 90%, which is very efficient. And the manure of the mealworms, of course, can be used as manure for the plants. But the idea was to grow these mealworms on uh, waste products from the plant production. And if that would work, and I'll start a small video, then you can see them moving. So they are thriving here. If that would work, then of course you could turn again a waste product into a high quality product that humans could eat, because the mealworms, they contain all 19 essential amino acids that humans need. So they could be a very, very healthy addition to your diet, and they can have up to 20% of protein content in their biomass. So it's also very easy to get a very healthy diet and to get to all your protein that you need on Mars or on the moon. So that would be a very, very good addition to the diet again. And of course, they are very easy to grow. Lotte experiment ended a couple of months ago, and we're on the verge to submitting the paper with the results in it. But I can already say that on average, we were able to have a biomass increase uh, in the mealworms. So they are growing. It is possible, but we still have to improve it because it's going slowly and we want to have it optimized again. So that's still work to do, but also, and that also has to be improved. They were growing on average, so the average weight increased, but the total weight of the mealworms still decreased. And we want to improve that because it's not enough to have an average growth of the mealworms. They also have to grow in total. Otherwise, it's of course not sustainable. And one of the reasons why that didn't happen was we think um, mealworms eat each other, especially when they going from one phase to the next phase. Their chitin panzer is weak then, and they eat each other. And if they have not enough fodder or not enough high quality fodder, then they will eat each other. And we think that is one of the reasons why the total weight did not increase. They ate partly each other. And that is also because we tested a lot of fodders, a lot of feed, and that was, of course, not optimal for them because it were waste products. So we are going to work on that as well. And even if we have to add some, for instance, grains or other products that we also can eat, it still may be beneficial for astronauts on Mars to eat the mealworms because of the nutritional value and because of the 19 essential amino acids that you have in your diet then. And to ensure, uh, I tried them, I tasted them. You can even buy them commercially already. Uh, the ones I tried were freeze dried and then baked, and they taste like a little bit like salty chips. They make also more or less the same sound, so I like them. So I think they could be a very, very good addition to your diet on Mars and on the moon as well. So that was my last slide. I added this as well. You can follow us, the research on YouTube, on Facebook, and also on Instagram and Twitter. So, and with that, I'm ending my presentation, and I would like to thank you very much for your attention. And I think we will go now to the Q&R, and I will stop sharing my presentation. So thank you for your attention.
I think first of all, Dr. Wamelink, there is a huge amount of applause for you. This, I'm sure
Thank you.
everybody in the audience is giving you a standing ovation on this. This is so inspiring. The only takeaway that I would take from this, except eating those lesser mealworms in the end, is that I would, if I was an agri student, this is going to be my area of interest. This is going to be my area of interest of my research. I would love to collaborate with you. And this is a message for all the students and the researchers who are actually seeing this. If you really have really not picked your area of interest, trust me, this is so promising. the kind of interest that we're already getting, specifically after the Chandrayaan success, trust me, there's going to be major grant around this. And if you can really work around growing food in harsher conditions, in conditions where there are no nutrients or anything, I'm like as tough as Mars and the moon, trust me, you can do anything on the earth. So if
Yes.
there's anything which you really want to pursue, this is something. I will be very glad to be working with Dr. Wamelink and Wageningen, you are in Netherlands. If we can come up with a boot camp for faculty members, researchers, or interested students to pursue this, and then come back and try and do on different other species and help him build a huge repository of all kinds of crops or whatever that is possible. Maybe there are new findings around this, but this really needs to go big. Thank you so much, Dr. Wamelink, for really, inspiring us and, taking us through a wonderful journey of your research around this. So,
Yes, and to add to that, if I may, thank you for your kind words, by the way. It makes me, well, a bit red. Thank you. But of course, one of the takeaway messages from this is it's not only exciting, but it has applications on Earth as well. And we already did that actually, because what we learned in these experiments, we also applied to see if we could grow crops on Sahara desert sand. And one of the other applications has been that on the space research we're doing in Wageningen is that we had a fully operational container on aquaponics in this case that went to the South Pole to feed the people, the researchers that are working over there, so they had fresh food. And of course, there are a lot of applications here on Earth,
exactly.
especially here in the Netherlands, because, well, that's not going too well at the moment.
Yeah, true. I mean, like, specifically when you mentioned that you really, I mean, like, people in Europe have really forgotten how to recycle your feces. And there's so many other probably practices which people have forgotten. There are a lot of learnings from things like this. And yes, true. When we do something like this, there are possibilities, which it opens a lot of possibilities on Earth as well in different conditions. Okay, so going forward, we don't have much time now. We only have 20 minutes for the Q&A. Please feel free to drop in your questions on the Q&A. Before that, I have a little poll. Meanwhile, Dr. Wamelink, if you can see the questions on the Q&A, you can start taking them up. And I'm
I'll
just launching
try,
the poll. Yeah, I'm just launching the
okay,
poll
I can
meanwhile.
see them.
Yes, and but you can start the questions.
Yes, well, I'll start just on the top for me.
Yeah.
From Macron, how does... Oh, sorry, it's gone. Now I see something else.
Okay, we just wanted to run a
I
quick
see now the
poll.
poll.
Yes,
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 I
we
see
just
the
want to run a
poll.
quick poll for just a minute.
Yeah,
So,
okay.
guys,
But
yeah,
the
please
questions are
go ahead.
back now. Yeah. How does gravity influence the plant metabolism? That is a very important question. And the honest question is, we only know partially the answer. And that's based on the work that's been done on the ISS. We know that plants will grow in microgravity, but even there, a lot is still unknown because, as far as I know, we still don't know if they grow as good and as fast as they would have on Earth. There have been some experiments on Earth I know of, but they are very limited. And one of the major things I think that came out of that is not about the metabolism, because I think nobody has done anything on that yet. So maybe something to pursue for you. But what I did look is at the growth of pollen to fertilize the plants. It's extremely important, as you can imagine, because otherwise you won't have seeds and not have maybe for some even a harvest. And they saw that under lower gravity, the growth of the pollen was slower, and also the pollen, the pollen tube was shorter, and that could influence, of course, the whole propagation of plant growth. But that's the only experiment I really know of. So there's lots more to do. Then to the second question of Madesh K. How might how might the lower gravity affect the growth and development of crops on the moon compared to Mars? Yeah, sorry, partly the same experiment. The only thing the same answer. The only thing uh, we speculated about, and of course it's not just speculation, is that we think that the crop growth, the height of the crops will be larger on the moon than on Mars due to the lower gravity. And we've discussed it with students and other people as well, because um, one of the key things in crop growth is gas exchange through the leaves. And is that going to be slower? That is one of the major questions. But up till now, there are no indications that that has happened on the ISS. So I suspect it will also not be a problem on Mars or the Moon. But there may be some differences between Mars and Moon. And also, what plants do is form tissue to make sure that they stand up. They make special tissue for that. Trees have cells, cambium, for instance, that they can grow that height and they are stable. And we think that under lower gravity, that may be less, and that may be that the plants will be more vulnerable. So if you touch them, that they will not be that strong. And that's something we want to look into, but also you have to be careful about. And maybe we need a little bit of wind just because of that to make the stems more firm. And that could also be influenced by the gravity. Then to the next question. Okay, thank you. Nice session, he says. Thank you. Uh, and then Lenovo about what was the basis of the selection of the plants. Yes, that has changed during time. But our first experiment, nobody ever tried this. So our first experiment was done as particularly on wild plants. And that was because we knew that those could stand more the harsh circumstances. So that was our selection criterion, and also if they could grow under nutrient poor circumstances. Because then maybe they would be able to grow. Now we know that they are able to grow, but we didn't know that back then. So that was very important. And of course, we wanted to have some crops, but the crops, and especially the crop species, was not that important at the beginning. Which crop species? And we chose garden cress and rye because we knew that they would germinate easily and germinate within 24 hours. So that we always would have had results, even if they could not grow on, that we would have had some germinating plants. So in the beginning, that was really important. But now it's totally different because we are now looking at only crops at the moment, although I have some oak trees somewhere standing as well as a side experiment to see if the trees will grow. Um, but we are looking only now at a balanced diet. I had some students that were looking into that, but NASA and ESA also do what is a balanced diet, what are the crops you have to grow for it. But it's not only about a balanced diet because I want it to be as efficient as possible. I can grow rice, for instance, but it's complicated and it takes time. Well, when I use potatoes, it's much more efficient. They need less space and they grow much farther, much quicker, and the harvest is also larger. And they take less space, which is important. So that is what we're trying to combine now. So to have a good harvest, but also to have it as efficient as possible, but it has also to be nutritional. And of course, the taste has to be great. It has to be a complete meal. And there is one special thing we are looking at. We're looking at plants that have a high um, content of vitamin E and vitamin C, and that is because of the radiation. Um, Vitamin E and C, when you eat it, can protect you from the harm that radiation may do, the cosmic radiation, because cosmic radiation normally gives you free radicals in your cells, so basically electrons that can damage other proteins, but also damage your DNA. And vitamin E and C can capture those free electrons, and by that, they are not harmful anymore. So by having a healthy diet with a lot of vitamin E and C, we hope to protect the astronauts from the effects of the poisonous radiation. So yes, that is how we look at it at the moment. And of course, we want to uh, uh, have them eat the total amount of uh, amino acids and proteins they need. And that is why also one of the major reasons why we're working on the mealworms at the moment. Um, well, the list of questions still is moving up and down. So
You can
I'm
pick the
just
best one in
scrolling,
interest
but
of time.
yes. I'll pick this one from Medish K. What role do hydroponics or aeroponics play in potential crop cultivation efforts on Mars and the Moon? I only talked about the regoliths, but I think they are important because we focus on the regoliths. But I think they are equally as important. So what I see is a greenhouse in a dome below ground still, but I see a greenhouse where you have three sections, where you have a hydroponic section, where you have an aeroponic section, and where you have a regolith section. And both you can use. For instance, tomatoes you can easily grow on hydroponics and also on aeroponics. But for potatoes, it's more complicated. It is possible, but it's complicated. And they grow very well in the regolith, I can say. And also the green beans grow very well. So I would have always those three options next to each other. I would also use algae. You could eat them, but you can also use them to manure the regolith. You can also use them to produce medicine. Um, so actually, you have four different types of growth chambers, and I think also it's important because in space research, space science, but also space travel, uh, it's very important to have backups. You always have backup systems. And that applies to a rocket, just for safety reasons, but that also applies to crop growth. So if you have a problem, for instance, with your aeroponics, then you still have your aquaponics and your regolith. If there is a problem with the regolith, you still have the aquaponics and the aeroponics. So I also see it as a backup system. We don't, as I said already before, we don't want to end up like Mark Watney in The Martian, stranded on Mars and having actually nothing to eat and grow your own crops. And when it fails, and it does in the movie, for who've seen it, then you're in trouble again. So you always have backup systems. So I think that is also very important. And using regoliths maybe look a bit complicated, but it also is easier to recycle organic matter and to recycle human feces. It makes it easier. I'll go to another question from Nilo. Thanks for the wonderful meeting. Thank you. Why don't we use synthetic and robotic worms? They do all the tasks of worms, but they don't have the possibility of contaminating Mars, and they won't attack plants as food. Well, yes, and I would like to take that a little bit broader because we know that there are already also robots that can do pollination. As far as I know at the moment, the pollen still has to be collected by people, but the pollination itself can be done by robots, by flying robots, so by drones, basically, in several different ways. So that is an option, and I think we will robotize a lot when we are on Mars. But at the moment, I still would like to go for the living ones, despite, as you say, just that, of course, the worms, they contain uh, bacteria in their gut. So you will bring that as well to Mars. And the question is, do you want that? But that also applies to us humans because we cannot live without the bacteria that are living in our gut. And that's a couple of kilos. We can be sterilized from the outside. That's no problem if we want to, but we still need them. So we will bring um, bacteria as well. And then, of course, you can say, well, why should we send humans in the first place? Just let us send robots that can do the job. But we believe that humans are still necessary at the moment. They can make different decisions than a robot does, and they can also make quicker decisions. When you are on Mars, of course, it takes time to communicate, and then humans at the site can make their own decisions. But I also think it is in the nature of humans to want to go to Mars and do the research on their own. And I'm still looking and believing in an more agriculture ecosystem and building that. But I totally agree with you. We have to be very careful and not let escape any living thing from our domes to the Martian surface. Although I think it's impossible for anything that we know now, and we know of the circumstances on Mars that they can live there. So it will die, but still it's a contamination and a pollution. So we have to be very careful. And actually, I do not know about synthetic robots that do exactly the same as the living ones do. I never heard of it, but I'll look into that. So thank you for that question as well. It's new for me. Let's go to this question about Mukesh Kumar. Your presentation is very nice on Mars. Thank you. Question about regolith for experiment of plant growth as competition Earth, Moon, and Mars. It's collected from these planets or artificially developed. I hope I understand the question correctly, but our idea is that at the start, we first bring everything we need because there's nothing there. So we are going to bring the bacteria we need. We're going to bring the fungi, the pollinators, the worms, and maybe we need more than just worms. Maybe we need more animals that live in the soil to bring. We are still not have that not complete. We're still working on that, but I hope we only have to bring it once, and that when we are there, we can use what we brought, and that it will sustain itself, so that it will be a sustainable agricultural ecosystem, and that we also can start breeding and plant breeding from the start over there, so that we get plants that are and also animals that are adjusted to the lower gravity and the Martian circumstances. That will take time, but I think we will bring the first batch with us, but then hopefully can work with the batch we brought, and that is it.
Thank you, Dr. Wieger. I think we still have time to take 1-2 more questions.
Yes, I will. So, I was already looking.
So can we take this question from Dr. Ravi Kumar?
Oh, sorry. Where is it?
Yeah.
Which question?
Kumar. Yeah. Just below Tarun Rathore.
Okay, sorry. I already scrolled, so I'm looking for it. Can you ask the question?
So what he says, what is the direct transitional implications of this research for what is this terrestrial agriculture? Because this is one question often asked by the policymakers, especially in the developing world.
Yes, excellent question. I only talked about Mars and on the moon, about the moon because I like it. But I think it has a lot of implications for earth agriculture as well, especially here in the Netherlands. I already hinted a bit about that agriculture here in the Netherlands. Although we produce a lot, we are one of the biggest producers in the world. It comes with a cost. And one of the biggest costs is at the moment that it's totally not sustainable. It's linear, and it's not circular. And by doing this research, we have to, because on Mars it has to be circular, and on the Moon it has to be circular, because there's nothing there. But it draws a lot of attention, and I think maybe that is one of the main results of the research. It's often in the media, and we always talk about sustainability and that it has to be circular, otherwise it is not possible. And we always hint on that we forgot about that here on Earth. So I think that is one of the main things. Take home messages is that it has to be circular, and that's not only on Mars. And we're doing that research, and we're looking at what is needed for such a circular ecosystem, and we've forgotten about that, especially here in the Netherlands. How important are the earthworms? By doing this research, we have the attention for the earthworms again, and that is also now back here in the Netherlands, that people say, well, we don't have enough earthworms in arable land. We don't have enough organic matter on arable land. And we're noticing that. And we're doing that research at the moment. And it's also important in relation to climate change that you have more organic matter in your soil, because there you store carbon. That you can even do that in arable land. But also important is, I think, what is needed for a circular agricultural ecosystem. And also is that if we get this going, and we want to build a dome here in the Wageningen campus, so to really try this out and to get it also there circular, so as a closed system, then in principle we have something, building and also the whole agricultural system that we can transfer to anywhere in the world. You could build it anywhere. You could build it in a desert. And the desert can still be desert, but you do it below ground. You have everything under control. The water, the amount of water that is needed will be limited because it has to be recycled. So if you close that system as well, you could do it anywhere on Earth. And if you can have it closed also for the water system, so recycle everything, then you could in principle do it with only a limited amount of water, which would help in deserts, for instance. So I think that is also a really important spin-off.
Thank you very much. I think Dr. Ravi got his answer. We can take one last question. Dr. Wieger, I think you can choose if you want, or I can read it for you.
Yeah, well,
yeah.
What I did not talk about, and I see a question about hypoxia condition on plant development. I would like to end with, well, it looks all very optimistic, maybe, the way I talk about it, but there are still issues. And one of the most important issues with Mars regolith, and we have it here on Earth as well, is the heavy metals and especially the perchlorate that is in the soil. And that could, especially the perchlorate, could be a showstopper on Mars because it's highly toxic for humans. If we breathe it, our lungs will be damaged in such a way that we die. But also the plants, it's highly toxic, and we have up to 2% of perchlorate in the soil. And there has to be a solution for that. And some student teams all over the world have been looking into that already. They came up with a sort of solution, but it's still not there. And we did our first experiments with that as well. There are bacteria that can break down perchlorate and turn it into chloride and oxygen. Well, the oxygen we can use, no problem there. And then we still have the chloride, which is, of course, also a problem. We know it as salt plains here all over the world. Some crops will grow in it, but most crops will not grow in it. So we still have to handle that. And we already did some experiments with that as well, where we grew saliconia, which can grow in very salt circumstances. It takes out the salt out of the soil, so we can amend the soil in such a way that the chloride is gone, but it can also done with heavy metals. You can use plant species that can take up heavy metals. For instance, quinoa can do it. It stores it in the lower leaves, and you can harvest those leaves, and it's also a way to get your metals out of the soil. But it also can make your soil more fertile and even more safe, and also your crops to be more safe to grow in the regolith. So there are still some major challenges where we have to work on. And I think that's a good thing to end with, because for the people who got enthusiastic, there's still a lot of work to do before we actually can do this on Mars or the Moon, or even apply it on Earth.
Dr. Wamelink, before we end, I think there is somebody who is actually wanting to put his question in front of you. And the question is it possible for rain-fed agriculture on Mars and Moon?
So rain-fed
agriculture.
Yeah, okay. Could you explain that a little bit? Because I'm trying to understand the question.
Yeah, because I think we will not have rain on the Moon and the Mars. So
yes, okay. Then I'll understand you correctly. Well, we're not going for rain, so we're not going for terraforming on Mars. I know some people do, but we are not. First of all, on the Moon there is not enough ice available to do something like that. There is no magnetic field. There's a big problem. Of course, Mars in the past did have water and clouds. That's now the general idea. But the problem is that Mars lost its magnetic field. And by losing its magnetic field, it lost its atmosphere, because you get ionized radiation in, and then it ionizes the molecules in your atmosphere, and then it can escape from Mars and go into space. That's actually what we think that happened on Mars. That's why Mars does not have an atmosphere anymore, because it has no magnetic field. So terraforming, in my opinion, on Mars will be impossible unless you restore that magnetic field. And I think that's something we don't know how to do yet. So for now, we still think that you have to do your agriculture indoors. And then, of course, you can have your own dripping system and giving water. So it will not be raining, but it will be like we do it in a greenhouse. And that's the easiest solution. And that's also doable if we are going in 2 years to the moon and in maybe 10 years to Mars. We can start that immediately. And then we can already feed the astronauts that are living there.
Absolutely. Thank you so much, Ravi Kumar Hossamani, for explaining that rain-fed is dryland agriculture. But I think we understand that since there is an absence of atmosphere or similar conditions on the Moon and the Mars, I think a controlled environment setup is only possible to begin with, and then there are possibilities. Dr. Wiener, Dr. Wieger Wamelink, maybe one more question if you want to, because people are really putting a lot of questions. I don't know if you have much time, maybe another 2 minutes. If you can just find some question. Meanwhile, I'll just share the poll result. What's really astonishing here is that every question, all the other questions are good enough. But the third question, which said, in fact, the second question, which said, which other country apart from India, USA, and China has landed on the moon? Surprisingly, the people are saying Germany, UK, and Japan, when the answer is none of this because it was USSR or Russia. So at least we should get that fact correct. But anyways, we're very proud of the fact that we have also landed on the moon. So yeah, one last question, Dr. Wamelink.
Yes, let's see. Or did you pick already one which is interesting? Because I have to scroll and look.
No worries. I think let's pick up the question of Dr. Angraj Ali, which says, which plant species as food will be the most ideal for Mars and Moon? I think you covered that.
I like that question very much because the crop we think will be most ideal up till now, what we've tested, of course, is the potato. And I like potatoes a lot. Huh? I'm Dutch, so we love our potatoes, especially when they are fried. Um, but there are reasons for it. We've been looking at rye, we've been looking at rice, other ones. First of all, when you are on Mars or the moon, you are in a harsh environment. So you need a lot of energy just to survive and do your work. So you need energetic food. Potato can serve that. Um, it still contains also a lot of other substances we need, minerals, things like that, although it's limited, so you cannot live on potatoes alone, be assured. But also when I look at the growing conditions, uh, I think we were now able to grow potatoes to harvest within 3 months in a greenhouse. But I think we can get that down to 10 weeks, which is very, very fast. And that also helps because the faster it grows to a harvest, the less space you need. And it is difficult on Mars and Moon, so everything that has to be built is complicated. So the less space you need for your greenhouse, the better it is. So that a potato is very efficient also helps. And what we've seen also is that the potato really likes to grow in the regoliths we have. And there are other crops that, well, they grow, but you can see it's not the best soil for them. The potatoes, marvelous. And of course, you need not much space in itself also to grow potatoes. They're not that big plants. It's easy to harvest. Uh, you have, of course, waste products, and that's a bit more complicated because for humans they are toxic, the waste products, but for many animals they are not, so they can be digested. So I would go for the potato, and the other one would be the strawberry. It's difficult to grow. It's still small. It's slow. So that's all against it. And also opposite what I said just before. But the strawberry has a lot of vitamin C and also vitamin E in it. And that can protect the astronauts from the poisonous radiation. And of course, it tastes great. That also helped because that's important for the well-being of the astronauts. So if I can choose two, then I would go for those two. But of course, it's not enough. You need more.
Great. I think we have many potato lovers across the world, so that's good news. I think Tarun, we are running out of time. Your question looks good. IIT Kanpur based. I am myself an alumnus of IIT Kanpur. So I would have loved this question. When IIT Kanpur based model on artificial cloud seeding. Dr. Wamelink, I mean, like he's requested, you want to take this question. The model model on artificial cloud seeding are lead to available to Mars planet. This is the third last question. I mean, like, in the second last question, Tarun Rathore. Third
Yes,
last.
but I'm afraid I cannot answer that question. It's out of my scope. So,
Okay.
but when we're talking clouds and water, what is interesting is that there is the idea that we use water, ice, to build a greenhouse. And if that would be possible, and it looks like it could be possible, then ice is very good in protecting you from radiation. It helps, especially UV, but also other radiation. And if that would work on Mars, then maybe you could have some rain getting back to the clouds, because it could drip off like you have an iglo. That could be an option, but I know they are thinking of that at the moment, and I'm not an engineer, so I cannot tell if that even is feasible, but I know they're thinking of that. So that could be of interest, but it's a bit out of my scope.
Okay. Thank you so much. I think we are done. We are taking most of the questions, and we already 10 minutes past the time. Thank you so much, Dr. Wieger Wamelink. It is a pleasure hosting you. Lots to learn. I'm like, there's so many people who are inspired. Everybody in the attendees list, I'm sure, is going to leave inspired today. Thank you so much for your time, and we really look forward to further more sessions from you. And have a good day.
Have a good day as well.
And good day, everyone who participated.
Good day. Yes, good day to everyone.
Thank you so much, sir.
Thank you very much. Thank you, everyone.
Okay. So, yes. Bye.
Bye-bye.